Hello everyone and welcome. On today's video, I am going to share what I have learned in Plankton Culture AQU4105. My name is Muhammad Anas. So, what is plankton? Plankton is actually a type of live feed. Most plankton can be found in freshwater and seawater. It's actually an organism that being carried by water flow. Meaning is they cannot swim against current. They act as a natural food with important nutritional linkage to secondary producer. The size can be from smallest 0.02 micrometer up to the biggest 2000 micrometer. Microalgae. Microalgae is actually a very tiny and small microscopic algae species found in freshwater and seawater. They are unicellular algae species that may either live singly or in colony. The purpose? They act as a green water. Is actually a food for zooplankton. So, the importance of microalgae. There are two microalgae, which is phytoplankton and zooplankton. For phytoplankton, they are primarily linked in food web. They are also able to produce oxygen and as a source of feed for zooplankton. The importance of zooplankton is source of food for fish larvae and as a very important food during earlier fish stage after yolk. So I'm going to share about the growth phase of microalgae. As you can see from my slide on the top right, there are 5 growth phases in microalgae. The first one is induction phase. During this phase, a little increase in density occurs. This phase happens when the algae culture is transferred from plate to a liquid culture. Second, the exponential phase. During this phase, the cell density increases rapidly or bloom. Third, the declining growth rate. The growth rate reaches its limit growth as the cell division slows down. Fourth, the stationary phase. During this period, the limiting factor and the growth rate are balanced, which resulting in relatively constant cell density. And finally, the death phase. The cell density decreases rapidly and the culture eventually collapses. They are incapable of sustaining life or growth. Okay, let's proceed to the other type of live feed that I have learned in this subject. Artemia. Artemia is also known as brine shrimp. They can produce cysts. The cysts have 3 layers, which is alveolar, is the hard layer, and then outer, which is also hard layer, and inner, which is a transparent layer. Copepod. Copepod is actually a small aquatic crustacean. The copepod can be found everywhere in water. They have a unique zigzag movement. Next is rotifer. Rotifer can produce in a very large quantity. They use for marine larvae species. They are very suitable for most fish mouths. So we move to the Cladocera. Cladocera is a freshwater zooplankton. There are various types of Cladocera, but the most important is Moina and Daphnia. Moina is actually a very relatively small than Daphnia, but Daphnia have a closed brood pouch compared to Moina, which is an open brood pouch. And lastly, Trochophores. Trochophores is a free-swimming larvae of marine annelid and mollusks. They are very small size, which is 50 micrometer, and suitable for fish larvae. Okay, I am going to share a technique that I have learned in this subject, which is upscaling method. So what is upscaling method anyway? Upscaling method is a process to extend the mass culture of microalgae. The aim for this process is to maximize the cell growth and the productivity through the optimization process of its parameter. This process helps to increase the volume of microalgae as well as to retain its quality for juvenile feeding purposes. So how to upscaling the microalgae culture? Okay, first, a low volume of collected water sample containing microalgae can be upscaled by transferring the water sample first into a larger culture vessel. Next, a 250 ml of fresh Conway media solution can be used as starter vessel to promote a higher cell density. And then, wait for a few days after inoculation, and the new culture vessel is placed under a strong illumination. The cell density will start to increase by observing the color intensity of the culture. After that, the culture will undergo exponential growth phase, as usually can be indicated by uniformly dark color intensity. It is recommended to transfer the culture to a larger vessel such as 1 liter container in order to increase the cell density. And finally, the process is repeated to subsequently larger culture vessel. In the end, usually in a form of fiberglass tank or massive raceway that will able to generate extremely high density of microalgae. So we have learned the upscaling method. But what is the advantages and disadvantages? Okay, for advantages, it can be continuously produced by a single starter culture. Next, the availability of green water. It is also good for a massive culture user. And for the disadvantages are, it need to be continuously harvested or maintained to avoid culture crash, and it is not suitable for small-scale culture. As for the conclusion, what have I learned from this subject? Okay, it is really important for aquaculturists, especially new students like me, to learn the plankton culture. Next, this subject highly press on the importance of making a live feed as earlier fish stage of feeding due to its nature. And finally, some of this technique can be done easily with a small guide, and it is practical for us to understand and learn about it.